Hello, I'm Matt Pettit, Chief Scientific Officer at Matcher Technologies, and I'm really excited to share some of the new features in the latest version of Matcher. The new version, 5.5, introduces new functionality available right now to complement the robust capabilities already available in Matcher. So let's dive into what's new. First, Matcher will now automatically notify you if a cycle is due to end before all of the necessary tasks in your standard operating procedures have been completed. This automated alert ensures that you have done what you said you were going to do for this cycle of treatment and that no critical steps have been overlooked. Second, not only is Matcher the only electronic witnessing system that effectively links donors to patients, but there's a new feature which highlights cases where a donor has been assigned to a patient cycle, making it clearly visible within your patient scheduler view. This will help to better communicate patient and donor relationships to all staff, improving visibility and further helping to reduce errors. And the third new feature I want to highlight is especially exciting because it will greatly enhance the service and support that we provide to our users. This feature, which is called Phone Home, allows Matcher to automatically notify our service and support team about potential technical issues, proactively helping to identify and address concerns before they become problems. Beyond these immediate updates, there's also more coming down the development pipeline, including hands-free mode for Pocket Matcher. But more on that soon. Of course, all these updates build on the powerful features introduced in version 5.4. So let's take a moment to revisit some of those key advancements that we developed in response to the evolving challenges of the IVF landscape. The first problem that we've tackled is that electronic witnessing systems are currently unable to integrate third-party barcodes, such as biopsy kits, into their witnessed procedures. And the question is often, if there's already a barcode there, can Matcher read it and identify it for witnessing or assign it to a patient? And the answer to that question is now yes. For example, assigning a genetics lab's existing non-Matcher barcode on a biopsy tube in a kit to a patient at the point of tubing and linking this unique barcode to the record in the patient's cryo inventory offers time savings and risk reduction to the clinics using pre-barcoded genetics kits. Once assigned to the patient, this non-Matcher barcode will be recognized in Matcher as belonging to the patient for all subsequent witnessing steps. And it doesn't stop at biopsy tubes. Scanning non-Matcher barcodes opens the doors to other possibilities, like assigning pre-barcoded blood tubes from LIMS systems, for example, matching barcoded time-lapse dishes like the ones used in EmbryoScope, assigning pre-barcoded cryopreserved gametes and embryos from other witnessing or cryo inventory systems, and even assigning a medicine at the point of dispensing are just a few examples. The next problem Matcher addresses relates to notification. Managers need to be notified about mistakes or non-conformances happening in real time. Our development team have taken this a step further and made this feature highly configurable in Matcher so they can effectively now trigger any event captured to automatically email a specific email address or even multiple email addresses to notify those staff members. This might be an alert to a quality manager that a mismatch has occurred, or it could be a more proactive alert, such as letting a product manager know that the product is about to reach its expiry date, for example. And this is a really fantastic tool because it not only sends up a flare if a non-conformance needs responding to, but it also provides proactive alerts to prevent a non-conformance from ever coming to bear. The next new feature deals with the problem that embryologists are not able to track a specific piece of patient barcoded labware and ensure that the same piece of labware is presented again at the correct point in the process. We've addressed this and we've called it Labware Tracker, and it allows the system to assign specific labware types to labware used in a procedure. To explain this, it's probably easier to use an example, and I'll use the example of sperm preparation. So when a sperm sample is collected, Matcher will recognise the barcode on the sperm container as the specific sperm container belonging to this patient. In the next step, when the sperm is moved out of the sperm container into a test tube for washing or sperm separation, for example, Matcher will expect to see the same sperm container, and if it doesn't, then the system will alert the user that there's a missing item in the procedure. Now, this forcing function ensures that embryologists don't just match the correct pre-assigned patient ID, but they're now able to track a specific piece of patient barcoded labware and ensure that the same piece of labware is presented again at the correct point in the process. This feature significantly enhances chain of custody, and it's what our customers have been asking for. The next problem was that the handheld barcode scanners lack some of the features and flexibility and speed needed to perform tasks without distractions. So now we've streamlined the Pocket Matcher witnessing interface and have additional functionality enhancing the user experience and assisting centres who prefer a Pocket Matcher only solution. This means that witnessing steps can be performed with lightning speed, which is safer for the embryos, and it makes processing time-critical steps like loading biopsy tubes and vitrification devices easier with less distractions. Another improvement has been addressed, and this is addressing the problem that user guides, onboarding and quick help support are not accessible immediately from within the application, which is when the guidance is needed the most. In Matcher version 5.4, we've now included an in-app user guide, onboarding and quick help support so that end users can access the guides and help immediately from within the application, which is when the guidance is most needed. This is especially good news for new users because it means that making the change from manual witnessing to electronic witnessing is much faster and it will run far smoother, which also reduces the risk of change management and reduces the amount of external support required, which saves further time and reduces distractions. And finally, currently there's no method available to dynamically change the freeze date of an embryo which has been frozen across consecutive freeze dates. Because embryos develop at different rates, our customers can't predict the specific freeze date of embryos vitrified in advance, and that means that creating the freeze in Matcher required the user to select one freeze date, which then had to be retrospectively edited for extended cultures and late developing blastocysts. But this is no longer a burden, and in Matcher version 5.4, the users are now able to dynamically change the freeze date of an embryo which has been frozen across consecutive freeze dates, and this includes on cryolabels. It's what our users have asked for, and it's what we've provided. To find out more, have a look at the What's New section on the website, or contact us for a demonstration of the new features. Thank you very much.